Okay, so there's really no one here. We could like all be having coffee together.
We could.
Let's do it.
I got a soda.
Let's do this.
So I want to apologize for teaching in the afternoon again next semester because it's not ideal. So for those of you taking 4 or 9, apologies that it's not at 11 8 class because I wish it was right now. There'll just be more soda to drink. Okay. There's like a whole line of people missing. One's here. Okay. Let's do this. Okay, so our business, someone said, what? The exam's not on 11/18. I was like, you are coming to lecture. So 11/20 is the exam. I believe I posted it on Blackboard that I was changing it. Tell your friends, or we could just see how many people show up on Monday. That could be fun too.
We can do that. Yeah, I like that.
Okay. So far, notice the font is getting smaller. Okay, everyone, let's come together. Should we stop, before we start, should we do like a stretching kind of yoga moment? Everyone get up, wake up, shake it off. Okay. Trying to get everyone focused. Yeah.
For the math, did you want us to know the specific ones, like how everything, or just the?
No, put them in categories. Put them in those categories. So I'll go through it, yeah. So I put maps just based on categories because there's so many, I don't want to list them all. But as you're going through them, put them all either in the category of involved in spacing organization, stabilizing, or destabilizing. Okay, so far, this is what you need to know. You guys should all know what you need to know because most of you have been through recitation and have seen the beauty of all these proteins and what they do. For those of you who are on Thursday's recitation, come see me if you have any questions afterwards because there's a lot of detail on this one. Okay, so we talked about all the key players and the major concepts and that we all know at this point. And then we started going through all the different accessory proteins, which is kind of what we're finishing at this point, right? There were the nucleating proteins, proteins that bind to monomer, proteins that sever, side-binding proteins. Some are maps, meaning they're associated with microtubules, and they might bind along the side or at the ends, but may have many different functions. So just categorize them based on them either being involved in spacing and organization, whether they're stabilizing, whether they're destabilizing. And for actin, we talked about cofilin and tropomyosin. One is destabilizing, one is stabilizing. Then the capping proteins, which is also, again, actin, CAPZ and tropomodulin. And then the cross-linking proteins is where we stopped. And that's where we'll begin today. And these are only for actin, the cross-linking proteins. Okay, so there are bundling proteins that will cross-link. Oh, I knew it was feeling bright. Is it better? So there are these bundling proteins that will cross-link actin filaments into a parallel array. So remember, actin has different functions, and you can kind of think of it as gel versus parallel, right? And you'll have your ARP or your ARP nucleated gel /web, and your parallel, which is nucleated by formants, right? And so you need to keep them in their structures because their structures directly relate to functions. So that's where these cross-linking proteins come in. So the bundling proteins, they'll cross-link actin filaments into their structure, and then they also determine what proteins might interact, and therefore can determine function. So for example, with myosin 2, motor protein, which we're about to learn about, it interacts with contractile bundles. And it can't interact with tightly packed actin filaments that are held together by fibrin. So here's two different types of cross-linking proteins. You have alpha-actinin and you have fibrin. And your alpha-actinin is going to create your contractile bundle, and there's a lot of space there, and that allows myosin to get in there and function. Whereas your parallel bundle is going to be, there's not enough space. So it's a much smaller cross-linking protein, and it's going to organize actin into a tighter packed bundle that won't allow myosin to it. So if I show you this and I ask you if myosin 2 functions in this, you have to say no. I won't do that, but it's good to understand structure relates to function. Okay? So the looser packing allows myosin to enter, making these bundles contractile. Okay? Good. No? Villan is another cross-linking protein, and that's these teeny little ones, and they're very small, and they pack actin even tighter, and similar structure to fibrin. See it there, and it cross-links the actin filaments and microvilli. So actually, if you overexpress villan in any sort of cells, in, for example, just cultured fibroblasts, you'll get increased numbers of microvilli. So they're very important in organizing actin into microvilli.
What's the purpose of the parallel bundles?
So in this case, so here, these are parallel bundles here. And so they're involved in the structure of the microvilli. So it's kind of a protrusion from a cell. Okay? So sometimes you'll have a contractile bundle. So you can have parallel bundle, you can have actin that forms in a web-like structure, and you can have actin that forms in a parallel structure. And based on how close the actin packs, there'll be different functions to the parallel structure. Okay? And so myosin can enter, because it's packed more loosely, like with the alpha-actinin, you're more apt to have contractile actin. Okay? Whereas here with the parallel bundle and also here, it's more forming a protrusion out of the cell. Okay? So these are our different cross-linking proteins. And the red right here, these are the actin binding domains. So you can imagine it would bind actin here, and it would bind actin here, or it would bind actin here, and it would bind actin here. So they're different sizes, and they're going to organize, based on the distance between the actin binding domains, it's going to organize actin into different bundles or conformations. So sometimes you'll have your actin binding domains separated by a long linker, and that's what forms your web or gel rather than bundle. So you can, if you compare filament to spectrin, you can imagine if your actin's binding here and here, it's going to cross over. And that's what it does, right? So here's your filament, and here's where your actin binding domain is, right? It's binding to actin. Going back to the concept of domains, remember it's the part of a protein that does something. So the actin binding domain here is binding actin, and its filament is shaped in such a way that it encourages formation of an actin web or gel that's associated with motility. Okay? So just think, with a cross-linking proteins, it can become as complicated or as simple as you like. And it's really about keeping an actin structure in a conformation so that it can do its job. And so if it needs to move, it's going to be in a certain kind of structure. If it needs to contract, it's going to be in a different kind of structure. If it's going to be a protrusion, it's going to be in a different kind of structure. And we've learned that, and these are these proteins that keep actin organized in such a way that it will maintain its structure. Okay? Oh, God. Okay, downward dog. I used to do yoga. Actually, so I was crazy about yoga at one point, long before having children. And children mess up everything. Like, all I could do was run now. Actually, no, I love my kids. Children who might listen to this one day. Everything exists forever on the internet, so I'm in trouble now. No, but like, I can't be held to a schedule, like a yoga schedule, right? But I used to go to Bikram yoga. Has anyone ever done Bikram yoga? The hot yoga that makes you want to die. Right. So I used to do that, and they had the 30-day challenge. And so like I'm like that kind of person who's like, oh, you're gonna challenge me? Well, you have to go every day of the week for 30 days. But if you miss one, then you got to double up. And so by the end, I had to do like 3 days in a row, double. I was like, I must have lost like 10 pounds of water weight from all that sweating. But anyways, so I got so addicted to that yoga but it was really expensive, and I was a poor student, so I started working at the yoga studio in exchange for free yoga, and I would clean their bathrooms. Yeah. Nothing is below. I was good at cleaning those bathrooms. Then I got pregnant with twins, and I thought, well, maybe I should stop cleaning the bathrooms at the yoga studio. Anyways, onwards. But there's no downward dog in Bikram yoga. I just want everyone to know that. Okay. So filament, okay, back to cross-linking proteins. There was no sense to any of that. I just needed to shake everyone out of the doldrums. We have a lot to get through, and I don't know how it's going to happen, but that's okay. So filament, right? So we just talked about filament. There it is again, right? Webs, motility. So filament is required for motility. Interestingly, some cancer cells, specifically malignant melanoma, lose expression of filament. So this is showing what the cell looks like without filament and with filament. See, that's very different in shape, right? And so since it's making that web and it's involved with motility, and it's involved in shape, right? So these have very distinct shape, whereas this is kind of this bloody mess, right? So would you think this is good or bad for the cancer?
I always thought that would be bad, because cancer cells don't need to be able to do.
Right, well, a successful cancer cell would want to be mobile, right? So it's bad for the cancer. Is it good or bad for the patient? Probably good. Well, so in actuality, when they're diagnosing melanoma, if they find that your flavor of melanoma has lost filament, it's actually a good prognosis because you're less likely to have like metastatic melanoma without filament. So for those of you, my 98-year-old grandfather just got diagnosed with melanoma. And I was kind of like, I know this is bad and maybe I'm cold, but I was kind of like, he's 98, right? So good for living in Florida. Good for him. Making it to 98 without melanoma. It's sad, but it's also like, how to get melanoma. 98 is a good time to get it. So I'm hoping that it's I'm hoping there's zero filament expression. Cut it out. Anyways. Okay. So actin cytoskeletal, as you would think, actin cytoskeletal structures need to attach to the plasma membrane. Does that make sense to you guys? We keep talking about how they localize to the plasma membrane, so that should mean that they're binding to something, right? Otherwise, why would they be there? So, right, they're affecting the shape of the plasma membrane, they're pushing up against the plasma membrane, so it makes sense that it would need to attach to it. Right, so there are these ERM proteins. This is bright. I don't know. Anyways, there are these ERM proteins, and they stand for ezrin, rhodixin, and moesin, 3 different proteins. And they're family of proteins that bind to the sides of actin filaments and to the cytoplasmic tail of transmembrane glycoproteins. Right, so they have 2 domains. One's an actin binding domain, and one is probably, I don't know, some kind of membrane protein binding domain. And they're regulated. So you can be in an inactive conformation and something can happen, such as phosphorylation, and you could become activated. And so you'll have a conformational change and be activated. And then your active ERM protein is going to bind to your glycoprotein, so to your membrane protein, and also bind to your actin. So that's how actin filaments can be recruited to the plasma membrane in certain locations. So this is one level of regulation of recruitment of actin to certain places. Yes.
Just going back to the point where you said it's needed for like motility. So I guess that's assuming like the treadmilling effect. I thought the treadmilling effect was a way for cells to really navigate in that. I don't remember us talking about filament.
Great. Okay, so you're talking about filament, you're talking about movement, you're talking about treadmilling, and you're trying to put it all together. Right? Okay. Yeah. So yeah, I hadn't introduced filament at that point, but yes, the treadmilling, and we'll start learning about it a little bit more. There are a lot of these accessory proteins that are important for moving the cell forward. And yes, treadmilling happens, but don't forget you also do need to maintain a structure in your actin web as you move forward. So you would thinking about filament, it's likely that binding is not permanent. It's probably reversible, regulated in some way. Yeah. And so then, so one thing is if you're, so if you need to move in some direction, you need to recruit all your actin to some side of the cell. Well, so here's one way to recruit actin to a certain location in the cell. Okay? And so these ERM proteins are targets of several RTKs. Do you guys remember what RTKs are? Receptor tyrosine kinases. So let that sink in for a minute. So right now you're thinking in your head, receptor tyrosine kinases. What were those? Who remembers what those were? Some kind of receptors that had kinase domains and they phosphorylated things. So some of the things that so they're activated by ligand binding, and then they phosphorylate themselves, and then they phosphorylate other things. And one of those targets would be ERM proteins. So we talked a lot about signaling and about activating gene expression, but also signaling also will activate cell movement, okay, or formation of cell structures. Okay, how are we feeling? I promise no more stories today. We'll have stories about motor proteins now. Okay, motor proteins move along cytoskeletal filaments. You know them all because you've drawn them in recitation or thinking about drawing them tomorrow. So I want you to organize them based on their track and their cargo when you start to think about them and their direction. So some factoids, they require ATP hydrolysis. There are many different kinds of motor proteins. Even there are many kinds of myosins, right? They can move along actin or microtubules, but they're specific. So myosin only moves along actin. Kinesin and dynein only move along microtubules. Some carry cargo to different locations in the cell. Some cause cytoskeletal filaments to slide against each other. They have different functions. Okay? So some general points. So all motor proteins will have a motor domain or a head region, which associates with whatever track it's walking on. So here your track is a microtubule, and so your motor domain, okay, I see it's showing it Your motor domain would be down here, okay? And the movement is dependent upon ATP binding and ATP hydrolysis. And so the proteins are cycling between binding to the track, whatever it is, nucleotide binding, nucleotide hydrolysis, some kind of conformational change, and filament release, and some kind of conformational change and filament binding. So I'm going to go through the exact mechanisms for the different motor proteins of this binds and this binds and this binds. I don't want you guys to get too bogged down with the detail. Just understand the basics of you get conformational change when you have ATP binding, you get another conformational change with ATP hydrolysis, and that's regulating the walking along the track. Okay? Get the basics. Yes?
That's what organelles can do the same thing as well.
Mhm.
So organelles and vesicles can both do actin and microtubules. Right?
Yep.
It's just that I guess the motor proteins are specific to what they do.
So, motor proteins, okay, so motor proteins have that motor domain, and that's going to be specific to the track. And then they're going to have their C-terminal domain, that's going to be specific to the cargo. And so that's where you determine what you're walking along and what you're binding to. So in some cases, you'll bind to a vesicle, in some cases, you'll bind to an organelle, in some cases, you'll bind to another cytoskeletal filament, depending on your motor protein.
But dynein
Dynein
will walk on actin.
will never walk on actin. Yeah, so those motor domains for dynein and kinesin are specific to microtubules. Yeah. So motor domain determines the identity of the track and the direction of the movement. And then the tail is determining the identity of the cargo. So your cargo here is your vesicle, or you could easily exchange that for an organelle. And your motor domain is binding to your microtubule. Okay? So members of the myosin superfamily, and I say superfamily because there are many members of this family, make up the actin-based motor proteins. So they are dimer, and so they have two heavy chains, and then they have the head domains. And so there's this coiled coil of these two alpha helices. And it'll bundle with other myosin. So if you're remembering learning in physiology about actin and myosin and muscle contraction, you probably remember seeing a lot of myosin heads, and it's because there are a lot of myosin motor proteins bundled together through the coiling of this coil domain. Okay? So this is actually probably what you've seen. So the myosin, this is myosin 2 that's involved in muscle contraction, and it assembles into the bipolar thick filament, and it's made up of many, many myosin 2 molecules. So that means that there are hundreds of myosin heads. Each one of these is a myosin head, and they're oriented in opposite directions. And they use ATP hydrolysis to walk to the plus end of the actin filament, and that's what's causing muscle contraction. And so that would be a close-up. So you'll have your myosin tail and then your myosin head, and they bundle together. So if you add ATP and actin together with myosin heads that are stuck to a glass slide, so here's a glass slide, and they've just immobilized myosin heads to it, and then thrown in actin and ATP. You can actually see the actin filaments gliding along the surface of the glass, so it'll actually move around. And if you think back to the paper we did on kinesin, that was one of the mobility tests that they did when they were trying to purify remember they were trying to purify an active kinesin, and then they were also looking at activity through different ways, and one was looking at the ability of kinesin to move around microtubules. So it was the same kind of assay. And so here, this is, oh God, sometimes these lights are so bright. Here you go. So you can actually see that these two different actin filaments are moving around on the glass. I believe I have a movie coming right up. Do I? It's coming up, I think. So ACP dependent movement of the myosin allows movement along the actin filament. Okay, so there's many different members of this family of motor proteins. These are all different myosins. They contained a conserved N-terminal motor domain. What does that mean? Conserved N-terminal motor domain. What is it? What does that mean about this domain? Yes. Huh? They're all pretty much the same. What were you saying over here?
They don't change.
They don't change. They're conserved. They're similar. It's at the N-terminus. It's a motor domain. They have the same function, right? And then they have a divergent C-terminal domain. What does this mean to you guys? I say the motor domain is conserved. The C-terminal domain is divergent. Why? Yes.
The C-terminal domain carries different cargo.
The C-terminal domain carries different cargo. Exactly. So, right. So here's the different types of myosins. Here's the conserved motor domains. And you can see in myosin 2, you get your contractile activity in muscle or non-muscle cells. Myosin 1 binds to actin in the plasma membrane. Myosin 6 is involved in vesicle and organelle transport. But notice they all have that same structure. Motor domain, and then some kind of C-terminal domain that has varying length. Okay? And then the other thing I forgot to point out is there's this neck or hinge region, and so that's kind of has to do with the length of the step that the motor protein is going to take. And so that'll also vary. So they have different lengths, right? So this is much longer, these are much shorter. So they're going to take different sized steps. So some interesting, can I see your hand going up? No? So some interesting myosin facts, just to keep things exciting. So it's not only present in muscle, hopefully you guys believe that by now. Who thought before taking this class that actin and myosin were all about muscle and that was it? Yeah. I think I may have thought that too before I heard about this. Yeah. Actin and myosin play a role in movement of the cell, just in general. They're found in protozoan cells and plants, but most of them are in eukaryotic cells. 9 out of 40 human myosins are expressed in hair cells of the inner ear. I don't know anything about physiology or the ear in general, and I found this to be pretty amazing when I learned this while reading this book. So mutations in 5 of these myosins are known to cause hereditary deafness. And so it's important for the construction and function of the stereocilia. Pretty cool, huh? All but one myosin move towards the plus end of actin. But most of these, there's no known function. There's really nothing known about. So myosin 1 binds actin plasma membrane, involved in cell surface protrusion. Myosin 2, contractile activity. So contractile activity is not only muscle contraction, but also the contractile activity that you see in cytokinesis and cell movement. Yes.
Is that really like a positive charge? Like I see a lot of cells saying something like
Plus n to minus n. Are you thinking charge? No, don't think charge. So thank you for bringing that up. So I just got a comment about the plus and minus n of actin, or even microtubules, right? And we're always talking about the plus and minus n. That really has to do with the end which most likely adds subunits. So your plus end is always going to add subunits, and your minus end is where most subunits are leaving from. And it's just a designation for your more quickly growing end, your plus end.
But a lot of times I see stuff that says like
Polar, well, polarity doesn't necessarily mean charge. It could just mean difference. So like you've got your bipolar, it means like two sides. Or you can have polarity in a cell, where you have a top and a bottom. So it's not always charged. Yeah. Good, but that I love those kind of questions because it's one of those basic things that probably a lot of people are confused about. Myosin 5 involved in vesicle and organelle transport. So don't limit yourself to movement of vesicles and organelles along microtubules. It's also, they're also moved by myosin along actin. But a lot of the other ones are not really known, not much is known. Okay, how do we feel about actin? Not actin, myosin. We feel good about actin? Let's not forget about actin. So now we're going to move on to kinesins. So these are microtubule motor proteins. You learned all about it because you read about how they discovered it and purified it and all that good stuff. So there are a lot of them also. It's not just one. It's a family. And many of them are involved in mitotic and meiotic spindle formation. So they also have conserved motor domains. And a similar, you're probably noticing, a very similar structure to myosin, right? You've got this globular head structure, so that's your motor domain. And then this middle coil region, which is also, it's a dimer, so it dimerizes through the coil region. And then here, the C-terminal domain is where you bind your cargo. Most walk to the plus end, except for one, kinesin 14. So we had this come up in recitation. Where is the plus end when we're talking about microtubules? Where is it located in the cell? In general, the plus end is in the center of the cell or near the periphery? Yes? Cell membrane, I heard someone say periphery. Yeah. Where's your minus end of microtubules? Centrosome, right. So if kinesins mostly walk to the plus end, they're usually walking from where to where? Huh? Centrosome out. Centrosome, so from the center to the outside. So centrosome to the plasma membrane. So if I was asking you a question, we talked about this just recently, about vesicular transport, then, and I said the vesicle is going, and so this is where you have to remember, even though the final is not cumulative, it's all cumulative. If I say in the secretory pathway, the vesicle is most likely to be carried by which motor protein? If a secretory vesicle is most likely to be carried by which motor protein? What would you say based on what you know? Kinesin, right? Because it walks to the plus end, right? You know what? Myosin is a possibility, right? Because there was myosin 5, which walks to the plus end, but if I ask specifically about microtubules, kinesin. Okay, so then there's dynin, another motor protein, but it's actually quite different in structure. So you could, this is always the structure I think of when I think motor proteins, right? You guys too? Do you have structures you think of? It's randomly, you're eating breakfast, thinking motor protein, and you think the structure. No? Well, dyneins actually look totally different. They have a very different structure. So they walk to the minus end, and they are the largest and fastest molecular motors. So there's cytoplasmic dynin, and there's also ciliary dynin. Your cytoplasmic dynin is important for vesicle trafficking, localization of the Golgi near the cell center, remember organelle movement. And then axonemal dynin, which is the ciliary dynin, which is involved in driving the beating of cilia and flagella. And I thought I had the structure. I'm sure it's coming. So myosin and kinesin, like I said, similar structure. So they both have the same structural core. They have this central ATP binding domain. So here again, we're talking domains. Hopefully everyone really understands domains at this point, right? One protein, this ATP binding domain, there's your ATP, there's your ATP, and they're similar. And they also have this lever arm. The only difference, notice this, so you've got your ATP binding domain, and then there's this other structure here. This is quite different, right? If you compare here, for those who can never see my pointer, to over here, right? This is your actin binding, and this is your microtubule binding. That's quite different. And that kind of explains they're different. They're both similar in that they function by using ATP hydrolysis to drive their movement, ATP binding and hydrolysis to drive their movement. But the structure over here is very different, which kind of explains how they have different tracks that they walk on. Interestingly, they're also somewhat structurally similar to RAS. Who remembers what RAS does? I heard someone say GTPase activity. Very good. What else do we remember? What do we remember about GTP binding proteins and how they work? Regulating something. I heard someone say molecular switches. What's similar, right, is that they have this GTP, GDP kind of activity where they're either on or off depending on whether or not they're GTP bound or GDP bound. And they have this conformational change that determines their function when they're on or off. That is based on that nucleotide binding. And so I guess it's not so surprising that another protein that requires some kind of nucleotide hydrolysis is also going to have a similar structure. So this, of course, suggests common evolutionary origin. Similar structures. Does everyone see the similar structures, or is it just my well-trained eyes? Yeah. I see pointing. We're gonna let her finish.
I think it's easy to see how they're similar and not just
Yep, you have to just stare at it for a while sometimes with structures. And when you start to see, I mean, people who do structural biology is kind of an amazing thing because people analyze structures and overlay structures of related proteins and then find how they're different and then they'll mutate them and see how the function changes. It's awesome. Right? Awesome? Okay. Okay, myosin. So this is the actual mechanism for myosin function. So it's using ATP hydrolysis induced conformational change to generate force. to move forward along the actin filament using a rocking lever arm. So that's this lever arm. So this, see the blue? This is what's moving. See how that rocks back and forth? It's in this facing the left and then it's facing the right. So the myosin head will be attached without the ATP, tightly bound to actin. Then ATP will come bind, and ATP binding will induce a conformational change. You got to think any time you have something binding to something, it's going to affect the structure, right? So ATP is going to bind, and it's going to cause a conformational change, and then release from actin. And then you'll get ATP hydrolysis, and then you'll get a change in conformation, because then again, you're going from ATP to ADP plus the pyrophosphate. And so that's the cocked conformation. And then there's the power stroke. And so then it's going to release the PI, and then it's going to bind, the head is going to bind further down. So you're going from binding this actin subunit to this actin subunit. And then you're going to get release of ADP. And so then you're going to be attached again. And so that's one move, that's one cycle of actin, of myosin movement. I have a movie.
Muscle myosin is a
So it's going
dimer
to talk,
with
and I'm
two
going to have a hard
identical
time with
motor
that part, but that's
heads
okay.
that act independently. Each myosin head has a catalytic core and an attached lever arm.
Okay, so here's your myosin head. There's your lever
A
arm.
coiled
Here's your
coil
actin.
rod ties the two heads
Here's your
together
coil
and
domain.
tethers them to the thick filament seen on top.
This is
Helical
actually
actin
muscle
filament
contraction,
is
so
shown
it's
at the
part of
bottom.
the thick filament, so that's a whole bunch
In the
of
beginning of the
myosin
movie,
all wound
The
together.
myosin heads contain bound ADP and phosphate and have weak affinity for
So
actin.
see, there's the two.
Once one of the heads
Head
docks
domains.
properly onto an actin subunit, phosphate is released.
So it binds to ATP,
Phosphate release
and then you get release of
strengthens
the
the binding
phosphate.
of the myosin head to actin, and also triggers the force-generating power stroke
And
that
so then
moves the actin filament.
that
ADP
rocking
then
lever arm is what
dissociates,
moves
and ATP binds
and
to
moves the
the
whole
empty
thing
nucleotide
forward.
binding
It
site,
releases ADP and
causing the
another
myosin
ATP to
head
bind.
to detach from the actin filament. On the detached head, ATP is hydrolyzed, which recocks the lever arm back
So
to
it's
its
that lever
pre-stroke
arm going
state.
back and forth. Well, but there's two.
Thus, like a spring, the arm stores the energy released by ATP hydrolysis, and the cycle can repeat. The actin filament does not slide back after being released by the motor head, because there are many other myosin molecules also attached to it, holding it under tension.
And so this is a part of a
The
very
swing of the lever arm can be directly observed on single myosin molecules. Here visualized by high-speed atomic force
So
microscopy.
this is from atomic force microscopy, and you can see that lever arm rocking right there. And so that's just the myosin itself. How is that? What does the other myosin head do? So you've got your bipolar thick filament, and on either side you have actin. So that's just showing one side. So that was for muscle contraction. I should find another movie which shows that. There was something dirty there. Oh, now it's not dirty. It's just ladies in bikinis. Why does that come up? Whatever. Because it's YouTube. Now, I know my search. I'm always like, ladies in bikinis. And someone else. Wouldn't that be embarrassing? Yep. Well, I'm going to stop myself right now. Okay. That was myosin. So then we've got kinesin, and it is also ATP dependent. So in this case, it's more of a walking motion. So rather than that one head along actin, here you can imagine 2 feet walking, and it kind of is like one swinging in front of the other. I have a movie there too. So in this case, you've got your ADP. This is your leading head. It's ADP bound, so it's loosely bound. And so then when it binds tightly, then ADP is released and ATP binds. That's right there. And the ATP binding causes a conformational change in the linker protein. And so again, that's this, and that's not the head part. The head part's binding to your track, but then it's this kind of lever arm linker protein that's linking between the head and the coil domain. And so it causes conformational change in the linker protein, which swings the rear motor protein forward. And then you replace with ATP, and then that drives forward the movement of the rear motor domain. Let me see, I'm pretty sure I have, yep, probably more movies of ladies in bikinis.
The motor protein kinesin is a dimer with 2 identical motor heads.
So
Each
here you're
head
going to
consists
show
of a
2 motor
catalytic
heads
core
because it's walking
and a neck
along microtubules.
In
And
the
those are
cell,
those linkers.
kinesins pull organelles along microtubule tracks. The organelle attaches to the other
So
end
here
of the
you've got
long
your
coil
motor
coil that holds the 2 motor heads
attached
together.
to your microtubules,
The
and your
organelle is
organelle
not
is
shown
going to be
here.
at your C terminus.
In solution, both kinesin heads contain tightly bound ADP and move randomly, driven by Brownian motion. When one of the 2 kinesin heads encounters a microtubule, it binds tightly.
So
Microtubule
when the
binding
head
causes
binds to the
ADP
microtubule,
to be
ADP is
from the
released and then
attached head.
ATP binds.
ATP then rapidly enters the empty nucleotide binding site. This nucleotide exchange triggers the neck linker to zipper onto the catalytic core.
So that exchange of
This
ADP
action
for
throws the
ATP
second head forward
makes this
and
linker
brings it near the
move
next binding
forward,
site on
which
the
kind of
microtubule.
swings your rear motor head forward.
The attached trailing head hydrolyzes the ATP and releases phosphate.
So
As
then you have the
neck
ATP
linker
hydrolysis,
unzippers from the
and
trailing
then
head,
you've got your
the leading
ATP
head
bound and
exchanges
loosely
its
bound.
nucleotide and zippers its neck linker onto the catalytic core, and the cycle repeats. In this way, kinesin dimers move processively, step by step, along the microtubule.
So it's really about that lever arm that's controlling the movement. Conformational changes driven by ATP binding or hydrolysis depending on the motor protein. Okay, so now there's no more ladies in bikinis, so it's not that embarrassing. Yeah.
What signals kinesin to move the muscles kind of be regulated by calcium? So what signals, what tells kinesin energy release?
Okay, so I just got a question, so you're going to make me think. I just got a question. What signals kinesin, or what regulates kinesin and tells it to move an organelle somewhere, right? Okay. So we're going to have to think for a minute. I want us all actually to think about this because I think we can do it together. Huh? Well, okay, so I'm hearing something about signaling. Okay, well, so let's think about vesicular transport because we've learned a lot about that. And so we know what directs, well, we kind of know what regulates membrane-membrane fusion, right? Because that's RABs and snares, right? But at nothing that has to do with interacting with motor proteins. So I wonder, but, okay, you're watching me think of my toes here. But organelles are moved also by motor proteins, suggesting there's some way, in some way they're close together, organelles are close together and vesicles are all close, they're all close to microtubules, right, because they're easily grabbed on by motor proteins and moved around. I'm not getting to the regulation yet. Does anyone have any thoughts or ideas about what regulates binding? It would be regulating binding, right, between the motor protein and the thing that's being moved. Yeah.
I mean,
I can get to the question.
I was thinking the expression of gene versus motor proteins. So if you allow something new to express a bunch of motor proteins that are specific to the gene movement,
So you're thinking just by increasing expression of the motor proteins,
specific.
there'll be increased numbers and they'll be present and able to move things. Yep.
Is it similar to like in muscles with troponin and tropomyosin? So it's just got something blocking it until it needs to.
Well, that's a very defined system, right? Because everything's fixed. Everything's fixed. So with troponin, but so that is a mechanism of regulation specific to actin and myosin in muscle. Absolutely. And we're going to learn that with troponin and tropomyosin. You guys are going to be like, snooze, I'm sorry. But that's very defined
Or could there be
because,
something like that that is blocking it?
well, so the reason, okay, I'm going to get you just 1 second. So the reason I'm pushing you away with that is because in muscle, it's very defined. Nothing's moving. It's all stuck there. And so you need to regulate access because what's preventing things from binding otherwise? So it is. It's truly about regulating access of myosin to the actin, the binding site on there, right? But with, I'll get you in 1 second, too. But with kinesin and dynein, there's such a large space and so much to move. And is it as simple as things having binding domains for in the cargo? I have no idea. It's a really great question. Yes.
That's what I was going to ask. Is there some kind of like binding domain within the organelle that can be regulated?
I mean, that would be a very obvious place to start. Whether there's a binding domain in the organelle or vesicle or whatever needs to be moved.
Well, can I, with the kinesin, is it always just like binding the microtubules, whether it has cargo or not, and walking along microtubules?
Is kinesin always binding to microtubules, whether there's cargo or not? No. No.
So,
And well, I have some ideas, but
if
no.
it's not binding, if it needs cargo to bind and actually move, then there would have to be some type of signaling sequence on the cargo.
So it's like, so again, something needs to regulate cargo binding, which needs to regulate interaction with the track, right? There's some level of regulation that must be there, right? It can't be random because things need to go where they're going places on purpose. I feel like this is a really nice stuff to teacher question, right? We've talked about it long enough that a lot of you guys have had a moment to think about it, right? So what regulates motor protein binding to cargo? Do not come back at me with actin, myosin, and muscle. That's not allowed. What regulates motor protein binding to cargo? The examples might be kinesin and dynein with vesicles or organelles. It's a very good question. I'm sure it's known, I'm sure people study it. And there's got to be some regulated binding domain or something like that. Okay, let's move on to dynein. You got 24 hours. Give me 24 hours to live. Give me a reference, or I give you nothing. And what we learned from the SRP question is if you can't find anything, it might be because it's not true or it's not new, and that too is an answer. So some people told me, I couldn't find anything, so I didn't send you anything. Well, people don't publish negative data. So you can't find anything. That's worth saying. Okay. Okay. So Dinans. Dinans couple ATP hydrolysis with microtubule binding and unbinding. So here you've got actually one head to generate movement through conformational change. So you have your microtubule and you've got your stalk, which has your motor domain. And then you've got your tail, which is going to attach to either cargo or some other microtubule. And what happens is addition of ATP and ATP hydrolysis results in movement of this motor head here. And so it actually results in this back and forth like ratcheting motion. And so the power stroke is driven by release of ADP and the pyrophosphate, and that causes this ring to rotate relative to the tail. And so I've got a movie. We'll see what else pops up. So you can actually see how it's kind of moving back and forth. And so that's the part that has the ATPase domain. That's the part down there that's going to be binding to the cargo. And this moving part, let's do it once more because I like that one. How do I go back? Oh. So you can imagine how that constant moving back and forth and binding and releasing is going to move along your microtubule. I believe this was constructed from electron microscopy. How do I get out of here? There we go. Okay. So let's do a little comparison. Motor protein similarities and differences. Similar. All of them have some sort of nucleotide hydrolysis that's associated with a conformational change that results in movement. And also what's similar is some small conformational changes that are happening. They're amplified by these lever arms. Well, so in myosin, it's the lever arm, it's the linker, and kinesin, and the stalk and diamond. So there's that web. You might have the ATP hydrolysis in this very small region, but it's attached to this longer region, and so this small movement is amplified to make a larger movement. How are they different? The conformation of the motor proteins is different in the different nucleotide bound states. So you can't say for all of them ATP bound means it's bound strongly to the track. Hydrolyzed, it's not. They're all different, and I don't want you to get bogged down with those details because that's not important to me. Just understand these kind of differences and similarities and the fact that you've got that conformational change that's driving the movement. So like I was saying before, they have different kinetics, which are determined by their structure and to their biological function. So you'll have these different lever arms in myosin. So myosin 2 versus myosin 5. And so they're going to walk a different length based on the size of their lever arm. So for myosin 2, it takes small steps before letting go. But it acts as part of the thick filament. So it needs to take a few steps and get out of the way because the thick filament is made up of many myosin heads. So if it's taking these very long steps, well, there's other myosin heads there. So small step, get out of the way. Myosin 5, this acts in vesicle transport, and it takes much longer steps. And spends more time bound to the actin filament, and it's acting movement over long periods, and it's acting alone versus it's part of this group of myosin heads. Right, so motor proteins are mediating transport of membrane-bound organelles. Nicotazole is a drug that depolymerizes microtubules. And so we already know they're involved in movement of organelle. And this is just proof. So in the absence of nicotazole, your microtubules, which are red, and your Golgi, which is green, your Golgi are all kind of where they should be. Remember, usually you have your ER and your Golgi all kind of centered around the nucleus. When you add nicotazole and you depolymerize all of your microtubules, what happens here? You guys want to take a minute? We only have another few minutes. Take a minute and think about what's happening to your Golgi. Talk to your friends. Wake up. It's polymerized microtubules. You have to ask yourself what that means. It's a drug. Yeah, nicotazole is a drug. That's, I believe, used in cancer treatment. It depolymerizes microtubules. That's actually just showing your
So, is it not to track that at all?
Yep.
What's the other one?
Well, so you so what? Yeah. So I just got a question that might lead me think that you guys might be overthinking this, as students love to do. So when you're looking at something like this, you always want to ask yourself, okay, what's the difference? So all you're looking at is red is your microtubules, and green is your Golgi. And you can see it's around the nucleus here, and here it's different. And so you have to ask yourself, when I depolymerize my microtubules by adding nicotazole, what's happening to my Golgi? So don't think what's happening to vesicles, what's happening. You can actually start thinking about what's going to happen to vesicles, but okay, what'd you guys come up with? Yep.
Maybe the cargo can transport from like one
So maybe cargo can't transport. Yep. Well, your Golgi stacks are not really Golgi stacks anymore. Everything's all over the place. Yep.
I thought maybe the motor proteins, like something with the recognition of the membrane bound organelles between differentiate between the fluid and stromal fluid, which is why it's going to
So you think that maybe the motor proteins didn't know what they should move and what they shouldn't move? Well, what's missing? What are the motor proteins walking on here? Microtubules. What are they walking on here? Nothing. Right. So you can say based on this that you were well. So does anyone want to add to that? So you could say that based on this, your motor proteins are keeping your Golgi where they need to be. Whereas if you have nothing to keep your Golgi where they need to be, they're going to just go everywhere. So it's about organization of your Golgi, right? So you can think of the movement of your organelles along the microtubule highway, right? So your minus ends are at the center of the cell, and your plus ends are at your plasma membrane. And we kind of talked about this. So I definitely want you to kind of think in this big picture kind of way as you're studying for the exam. What motor protein would be involved in transport of this or transport of that? And if you understand where your minus ends are and where your plus ends are and which direction each motor protein walks in, you should have no problem associating that with cell processes. Yeah.
Is there like any benefit for having diamine move toward a negative like why is diamine so much faster?
Oh. So dynin goes to the minus end and kinesin goes to the plus end. The dynin goes fast. What's the benefit of that? That's the question, right? It's a good one. What do you guys think? What's going to be moving from outwards to inwards? Yes. So to keep things from going out. So the question really though is dynin moves inwards. So oh, so when you're talking about retrograde transport? Okay, so that's one thing that needs to go back. But you're asking about the speed, right? The dynin's so fast. Well, that might have to do the speed. I don't know. We have to think about that one. I'm going to leave you guys. We're going to finish now. And I'm going to leave you guys with that question. And we're going to start again with that on Monday. When we talk about good question.
I'm making a note for myself.
Remember with this slide, actin filament in the test tube versus actin filament in the cell, and the actin filament in the test tube was more static and persisted longer than actin in the cell. It was more dynamic, and the whole reason was that you didn't have things breaking it down in the cell.
Yeah, well,
I mean in the test tube.
yeah, in the test tube, if you only put actin in, it's only going to be governed by.
But you were
critical
talking about
concentration.
not actin monomers. You were talking about actin filament.
No, I was
right?
talking about if you throw actin, period. Well, filament or monomer it doesn't make a difference, right?
Right.
Because it's going to be fluctuating back and forth based on critical concentration,
Right.
and that leads to hydrolysis, right?
Right.
So,
So why is it persisting in this? Why is it persisting in the tube longer than it does in the cell?
well, so you can think of it either way. So in the cell, in the tube, it's going to be based on this 1 micromolar critical concentration.
It's just going to stay there.
But in the cell, you've got things like thymosin, and
okay.
you've got things like.
I thought I was reading the slide backwards. when I
No, but you
read
can
the
actually
slide.
probably think of it both ways,
Yeah.
right? Depending on what I tell you, you should think about.
Yeah.
Because
Depending
if
on
I
what
say
you say,
muscle
what's
cell,
happening in the cell.
If I say muscle cell, it's a different story. You're very stable in a muscle cell because you've got your CAP-C and
Maybe you
your,
don't have
your
that
triple
in
modulator.
Maybe you don't have that in the
Well,
test tube.
the test tube, you don't have anything but
Yeah.
actin.
Right, just
Yeah.
the amount of actin you put in there.
Yep.
You just
Yep.
Hello.
Hey.
I forgot to email you Monday.